So yeah, hi, my name is Rich Abdill. I'm the computational lab manager in Ron Blackman's lab at the University of Chicago. And I'm honored to be here to talk to you about our experience building the Human Microbiome Compendium using publicly available sequencing data. I'm also excited to be in this special session, so I've changed the talk around to be more about our methods than our results, but I promise there are results in the preprint. There's a link at the end. There used to be a link on this slide, and it seems to have disappeared. But yeah, I'm a biologist, I'm a microbiome researcher, but today I'm talking to you as a data janitor. So I'm particularly interested in the human gut microbiome, which is the functional capacity of the organisms that colonize the gut. They do a lot to help us digest things, particularly dietary fiber. The rough estimate is that somewhere between 5 and 10% of our energy balance comes from microbial fermentation that's happening in there. We give our nice little friends in there polysaccharides, and they give us short-chain fatty acids that we can burn up for energy. They give us vitamin K, a couple B vitamins that we can't get from anywhere else. So they're really helpful. But when things get disrupted, they can also cause problems. They've been linked to, if you Google the human gut microbiome, a million different conditions. Some of them are probably real, some of them are probably not. Yeah, they can interfere with host signaling. They can mediate the severity of flare-ups of IBD or irritable bowel syndrome. There's a lot of things that can happen, and most of the time, it's not one type of bacteria that's to blame. Sometimes, yes, you have an ulcer and we can blame Fusobacterium or something like that, but most of the time, it's these community dynamics that are to blame. It's alpha diversity. It's how are these different bacteria interacting with each other. Some of them are feeding each other. Some of them may be working really hard to crowd out other ones. So having a holistic understanding of the gut microbiome means understanding what ecological processes are kind of governing these little systems. And the main way that we at least get a census of which bacteria are in there is using different sequencing assays. And at that point, you'll have a list of samples and then how many or what proportion of each kind of bacteria are in each sample. And the problem at that point is power. If you have two groups and you're trying to figure out what's different between them, you might have 40 study subjects to figure out 900 different taxa that they share between the two of them, which is not great news. That's a really wide table that's really shallow. So even if you're looking for something with a big effect size, it can be really challenging to figure out where it's at. Luckily, I guess, there's probably a lot of irrelevant information in those 900 variables you're wrestling with, or at least redundant information. And there are lots of different ways to try to tease out what those might be. And one of the more promising avenues has been machine learning, which there's been no shortage of talks at the last few days. And particularly in the gene expression space, they've been having a lot of success using systems where they're using tens of thousands of samples from these public repositories to train models that can learn these kind of big picture patterns that we can see across lots of different conditions and lots of different situations. And that can either highlight which variables are important or extract these new kind of latent variables that are linked to lots of different genes or lots of different taxa, hopefully. And that can solve some of those problems. And the issue in the human gut microbiome space in particular is that there wasn't a comparable resource like something like Recount3 or the Cancer Genome Atlas, where the data's there and we just have to figure out how we can use it to train a model. There wasn't the training data. So we wanted to try to put something together. And so the question at that point was, what kind of data should we be looking for? There are the two main ways of kind of surveying the gut microbiome is shotgun metagenomic sequencing, which is where you extract all the bacterial DNA, you sequence what you can, and then you get a nice picture of at least part of the genomes for lots of the taxa that are in there. The other way, and the older approach is 16S rRNA amplicon sequencing, which is focused on one, or in an individual project, they'd be focused on one region of one gene that's common to all bacteria. And we can use little variations in the sequence to infer which species that read came from, or at least which genera, hopefully. And there are pros and cons to each, but one of the things that we were aware of going in was something we ran into in a previous study where it's on this graph here. We looked at all the biosamples that were submitted to the NCBI databases. So the National Center for Biotechnology Information, they're the ones who run the Sequence Read Archive and Biosample and Bioproject and GEO and all these miraculous databases where everyone is forced to deposit their data now. And it also pulls in information from a consortium of other repositories in the EU and in Japan. So this is almost 400,000 samples in this plot, and we basically used the metadata to annotate them all with their country of origin and then summarize things at the regional level. And this is what we found. The x-axis is time, and the y-axis is cumulatively the number of samples from these regions. And then the little bar on the right side uses the same colors, but it's each region's share of the world population. So you can see it's not exactly an even sampling. Even pulling in data from all these different repositories, Europe and North America it represents the wide majority of publicly available microbiome data that we can get to. And so we had to keep that in mind. So we went back a couple of years later when we were starting the compendium project and did the same thing again, but split out amplicon sequencing from the metagenomic sequencing. And we found that shotgun data is even more heavily skewed toward like what people will call like the Western microbiome. It's the global North. Wealthy countries get shotgun sequencing, and everyone else is still using 16S. And it's not to say that there aren't 16S studies going on now. There's 16S studies going on here probably. It was just that if we wanted to understand as much global variation in the human gut microbiome as we could, and assuming that microbiomes in different places tend to look different, then 16S was the only way to go, at least at first, because that's where that's the type of data where we could get the most different kinds. And it helps that it's also the most data sets. But once we had that figured out, that meant we could get started. And this is how we started. The graph won't always look this silly. This is just the first few squares, but it was basically just pulling all the extra metadata that we could find in these databases about these 16S samples and just organizing them all in a little SQLite database that was just running on my laptop. It wasn't anything exotic. And once we had that sample information, we could start processing them one project at a time. And the pipeline that we use is a very ordinary pipeline using Data 2. It uses as many of the kind of conventional approaches as we could put together. And it's all automated using SnakeMake. Nextflow got mentioned earlier. If you're not familiar with workflow managers like that, the basic idea is that you can define, to process this project, step 1 is this command, then step 2 is this other command, and then for every sample you run steps 3, 4, 5, and it can, it has different little helpers within it to keep track of progress through that pipeline for all the different samples. And that's great once you can get it to run. That's great for processing a single project. But by the time we got to this point, we had about 800 projects that we wanted to process. And so it wasn't practical to, just do that by hand, though we did try. And we needed some automation for our automation, basically. And that's where we came in with what I'm calling the compendium manager, which is just a little Python application that runs on a cluster. And when you launch it, it goes into the database, finds an unprocessed project, and launches a SnakeMake pipeline to process it. The only input for SnakeMake is the ID of the bio project and just a list of the samples. And then SnakeMake can go in and download all that data from the same NCBI sources that we got the metadata from and start processing each project separately. And then SnakeMake, because most of those samples can, for the most part, be processed independently, is launching it within each project also. So it's basically using Slurm or one of these other schedulers. You can say, start these 3 projects, and then however many resources you can get hold of at that time, that's how many samples are being processed. And then the last step is to pull together all those samples from the project into a couple of results files and some QC reports. And that all just gets collected into text files. And then the last step in the SnakeMake pipeline is to call the compendium manager back and say, there's a project that needs to be evaluated. And then it can go in and load those QC reports and figure out what happened. If it didn't work, maybe the reads didn't merge, or there's lots of chimeric reads for some reason, it can change the parameters of the pipeline and try again. If it already tried again for that project, it can record what the problem was and then just kind of discard that. But if it works, the next step is to take those results, all that count data basically, and put it back into the database and then delete all those files and make space on the cluster because we don't have that much. And the connection that really made the difference was this one. Once a project is resolved in one way or another, the next thing that happens is the compendium manager goes back to that database, finds another unprocessed project, and starts processing that one. So you can kind of get the process started, and then hopefully you can let it ride and check a couple weeks later and see how your compendium is going along. And this is what we found for version 1, at least. We ended up with about 168,000 samples processed from 481 projects. And the top left graph here is a PCA of the composition of those samples colored by what region it's from. And then the others are just a heat map or heat maps for each region that shows the density of points from each region there, because there's a lot of overlap and it's kind of hard to tell. And we can see that at least in general, microbiomes from a given region tend to cluster in these little areas, even when we're just looking at these first 2 dimensions. And so then we can really dig into that data and see what kind of variation happens within a region, or which taxa are most useful in differentiating samples from different regions, or just big picture, which taxa always show up, which show up only in one region, and things like that. And I think that's really exciting, even before we get into getting to use this as training data for some fancy neural network. And there's plenty of other results in our paper if you want to check it out. Hopefully there'll be a published version at some point, but we at least have a preprint. But my title slide promised lessons, and I don't think I have too much time left, so I'll go through them quickly. There's only 4, and they are not super controversial. So the first one is just that it can be really helpful to keep in mind how all these different steps are going to be coordinated. Sometimes it's just a sad grad student running bash scripts, but it's nice to have things communicating with each other. And that's something we kind of learned slowly through the process was, like the compendium manager can launch a snake-make pipeline. That's really handy, but the part that really made the difference was that the snake-make pipeline calls the compendium manager back at the end. And that's the connection that turns it from like a nice little command line tool into an automated system where you provide the parameters and tell it what to do, but then it can go and kind of take care of the rest, and knowing that it's more consistently applying your kind of thresholds and your filters than you'd be doing if you could do it by hand. But doing it by hand can be really helpful. Number 2 is that manually processing the data can be helpful in finding stuff that would otherwise kind of get papered over. So just weird things where the project would probably have otherwise been discarded because it seemed like non-applicable data. But since we were doing it by hand, we realized, oh, we need to add this extra step in the pipeline, and it can rescue these 300 samples, and we can add them in also. But automation can also help. There are some problems where you can only find them manually because it's some nuanced thing that you wouldn't notice otherwise. But automation can help everything go faster, including finding all these problems. So it's the project that only has reverse reads for some reason, or the project that combined human samples and mouse samples and soil samples and called them all human gut metagenome. So some of this stuff is only found because you can get all that data moving through. And number 4, and I promise it's the last one, is expect to start over. I don't mean this to be discouraging. I personally was discouraged, but basically there are some things where you're not going to be able to go back and fix it later. Some problems you run into it for the first time, you do a manual fix, and then it's automated from there. But if you're 30,000 samples in and you realize you missed a QC step, you're fixing the pipeline and starting over. And that can be useful to keep in mind just for planning a project timeline or trying to budget computational time that you have. And so this is just a quick summary of those lessons. I don't think there's anything controversial, but these would have been really convenient to learn on purpose instead of by accident for us. So I'm hoping they're helpful for you. And that's it. I promise I'm done talking. I just wanted to thank my supervisor, Ron Bleckman, and my first author, Samantha Graham. And I'm happy to answer any questions in my negative 10 seconds remaining.